Hi, Catherine. Can you hear me okay? Hi, Catherine. Oh, hi. Okay, great. Sorry, today we have to do on Zoom. Are you at office, Catherine? Yeah. I just want to make sure. Are you at home or are you at office? No, you're home? Okay. So I think office is trying to set up. There are some student in the office. I'm actually not at office either. So give me 1 sec. We'll get started. Yeah. Okay. Have you taken the test yet, Catherine? Yeah. So okay, you did the free FRQ. Okay, so look over the multiple choice right now while you're waiting. Let me make sure the office has this set up.
Hello. Oh, cannot hear. Is the audio on? You guys don't have to know how to control the game. I know. Okay, that's fine. Bear with me a little bit. What do you need? I need the audio. Oh, just use the The computer. It seems just so so. Yes. Sound. Okay. First one. This one.
Hey guys.
Okay. Oh, that's not letting me move. There it is. This one. Hello, Daniel.
Hi.
Can you hear us? Okay. We are trying to we're trying to make you louder
Okay. If I speak like this, is it louder?
a little bit.
A lot. Uh,
Zoom settings. So, this one? Oh, the Zoom link. It's not letting me close. Okay, this. Yeah. Okay. This one. Okay. Can you hear us? Daniel, can you say something?
Hi.
Yay! Okay.
Not too loud.
No,
Sorry,
it's
guys.
good. You're
All right.
coming from the speaker. Yeah.
Okay. I'll try to speak clearly. Hi, guys. Thanks for checking in. It's supposed to be in person today, but something came up, so we have to do Zoom. Okay. Again, yeah. So this is weird because we're doing Zoom on this way. Can you guys see the whiteboard? I mean, can you guys see the free response
Oh.
questions?
Oh.
Um, Catherine?
No.
I mean, Catherine can, I
Yeah.
know. How about Office? Can you see
Oh.
what I'm sharing? Angela,
Yes.
can you see I'm my screen?
Yeah, hold on. Sorry, I have everything opened up. Let me.
Yeah, can you see my screen? Because I don't think you guys are seeing my screen.
Oh. Yeah, we can see it.
You can see it? Yeah. Do you see this experiment here?
Let me.
Okay, great. And just you might want to turn off the lights a little bit so it's brighter. That's what I usually do on Sundays.
Oh.
All right. Well, the test itself is not too bad. This is actually one of the earlier AP that is released. And again, it has questions no longer on the new AP. There's no more fill in. So don't worry about the fill in. And then there's only a 53 multiple choice question, I think. Um, you're going to get 60. Yeah. And then the free response, there's 8. But the new ones are, again, 6. Okay. So let's take a look at this. Yeah. Thanks for taking the entire test. I know it is not easy to do, but we're almost there. Yeah. So here's the question. So let's take a look at this one. Okay. And if you have questions, feel free just to let me know. I may be able to hear you, may not. Okay. So is everything okay now, Angela?
Yes,
Okay.
I think
All right.
so.
All right, thanks, guys. So this one here is one of the lab we need to know. This is called a choice chamber. Yeah, choice. So you have a choice. In this case, we're gonna have fruit flies inserted in the middle, and the two choices are the left side is glucose soaked cotton, and the right side is dry cotton. So the idea is that if you insert 60 flies in the middle of the chamber, well, we're going to figure out which side the fruit fly prefers, right? So they do this with food source. So you can see that the left side is glucose. And they do this with water, humidity. Do they prefer water or not? They do this with light and dark. And they do this with bright and dark, right? Hot and cold. So this is called a choice chamber. So this is the investigation. Okay, a covered choice chamber used to test whether the spatial distribution of light is affected by the substance phase at one end. Yeah. So here we have a cotton bowl that is soaked with 10% glucose solution. So that would be 10 parts glucose. The rest of them is water. So you put this on one end, and the other end is dry cotton. Okay, the position of the fly are observed and obtained and recorded every 10 minutes. Now, there's some problem with this setup right away because if you have only 10% glucose, that means 90% of it is water, then we are not sure, are we really testing for the preference of glucose or are we testing for the preference of water? There's two variable that is actually being tested here. So that's the problem with the setup. So either it's 100% sugar or 100% water. When you have a glucose water mixture and you got a whole bunch of flies surrounding the glucose soaked cotton, we are not sure whether the flies are there for water or the flies are there for glucose. So that is one thing you have to improve as an experimenter, right? Something is wrong, okay? So you have two variables that is being investigated. Okay, so the question here is predict the distribution of flies in the chamber after 10 minutes. Yeah. Now you have, we will have to self-grade right now. So your prediction is common sense will tell you flies will prefer the glucose soaked cotton, right? Justify because glucose is a source of nutrition. Or you can say water is favorable, right? So that's it. You don't have to say how many flies. So you just have to say flies prefer the glucose soaked cotton. Okay. And your justification would be because glucose is a favorable source of nutrients. Okay. Did you guys put anybody put a dry cotton? The contrarian. So if you did, you had to justify it, right? So the prediction doesn't have to be always the common sense. Yeah. But unfortunately, in this example, you will have no points if you chose the dry cotton. All right. So please give yourself 1 or 2 points here. We're going to start with free response before we go to the multiple choice. Yeah. All right. So let's look at part B. So B here, okay, so I'm going to split the screen a little bit, is asking us to propose one specific improvement on the experiment, the experimental design, right? And explain a little bit how the modification will make it better. So I'm going to start with the experimental control. So this is what I was commenting, yeah? Right now, the control here seems to be the dry cotton. Well, that's not a very good control if you're trying to test, okay? So what exactly is the experiment trying to test here? And you can see it's this. They're trying to test whether, okay, so fruit behavior, you're trying to test the glucose, right? This is the substance that they're talking about here. So if I want to improve the control, I would have to use a control that is not dry cotton, but it should be a water soaked wet cotton, for example. Yeah. Okay. So for part B, if I had chosen water soaked cotton, so use water soaked cotton instead of dry cotton, that would be a big plus. Okay. So how do you explain it though? Because this is 1 point for your proposal, right? But now you got to explain it how this will affect the experiment. So to eliminate water, okay? To eliminate water is what? To eliminate water as a preference for fruit fly behavior, right? To eliminate water as a variable for fly, I'm gonna say fruit fly behavior, right? Okay, so that would be the justification. Okay, so that way we know if the fruit flies are moving to the left side of the choice chamber, they are there for glucose, not for water, because there's water on the in my control. So that's important to say. Now there might be other ways you can talk about it. You can so this one you can say that to ensure to eliminate this one, or okay, to ensure that glucose, not water, is the reason for flies movement, right? So you can also say that. I just want you to show you the different ways you can talk about it. Okay. Alrighty. Okay. Now that would be the control. So control specifically is this. Now don't forget, there's always two type of control. So this is just additional. Yeah. We have the positive control and also the negative control. So sometimes they force you to talk about, is this a positive control or is it a negative control? So positive control is a control that would produce the desired experimental result. So this will produce the desired experimental result, and negative control will not will not produce the desired result. Yeah. So the two type of control. So maybe a very quick example. Okay, let's say you test drugs or vaccines, right? Remember how the COVID vaccine was tested before they were released for every one of us to get shots? A placebo, placebo is a negative control. Placebo is just a sugar pill or sugar solution, not the vaccine, yeah? Instead of the vaccine, right? So that's usually the negative control, because if you just take in the sugar solution or sugar pill, you're not going to produce the desired results, right? That could be an example of a negative control. Okay, so there's positive and negative control. We'll talk about more when we see them. Now, that is 2 points right there, yeah? And now what about the experimental factors? So since we're talking about the food preference for the fruit fly, which is glucose, the experimental factors, which are always the same things. What are the experimental factors for any experiment? So that would be temperature. That could be the amount of light. Sound might affect the fruit fly movement, right? You cannot have one end that is very loud and scare the heck out of all the flies, or one end is very hot. It's like cooking the flies to death, right? So all these must be held constant. And if you notice that during the question, when we read this, if you read this another time, it mentions nothing about the experiment, the experimental factors. We don't know if the two sides have constant amount of sunlight, whether they are the same temperature, whether they're soundproof, right? So there's no mention. So how do you make it better? Well, fix, okay, fix the temperature or sound or light. So you can talk about any of them, okay? So these are some of the sound. You don't have to talk about all of them. So how the modification, so want to keep all variables. Okay, so let's see, we can use keep all variables other than glucose constant, yeah. Okay, so example will be keep light intensity for both chamber at ambient level, not too bright, not too dark, right? Throughout the experiment, okay? Throughout the experiment. So you can also talk about other things. So it's not just about, I use example for light, right? So you can talk about, again, temperatures. You can talk about, yeah, you can talk about sound. Basically, that's it, okay? So keep everything else other than whatever you're trying to test constant, so that they are not contributing to any fruit fly behavior. Okay, so so this is the proposal. So now we're gonna explain, right? So that the light intensity does not affect flies movement between chambers, right? So can see that. Okay, so anyway, so like this. Alrighty, that's part B. So because there's two bold terms and two bullet, this is 4 points right here. Yeah, 4 points. Alrighty. Is that okay so far? So far, pretty easy. So this is one of the choice chamber lab. Okay, now let's look at this now. The experiment described above is repeated with ripe banana on one end and unripe banana at the other end. Again, is food, yeah? So this is good because the ripe banana probably has more scent, odorous molecules that flies can detect, right? And by the way, if you don't know, flies they taste food with their feet. It's very gross, yeah. So they actually land on the molecules. Their receptors, chemoreceptors on the foot, and then they will taste with the foot to see if they like it. Okay. So that's why if you go picnic, make sure you cover your food because the flies will land around whatever they see, dog poo, for example, and then they will come and visit your hot dogs or salsa, right? So that's why we try to keep it isolated. So that's how they taste. So this is the chemical receptors flies can detect. So then again, the position of the flies are recorded every 10 minutes and the position after 1 minute and after 10 minutes are shown. So this is our data. So they don't want you to actually do the experiment, but if you had done an experiment, you got this data. So you can see that the flies were inserted in the middle. So that's why there's 3 regions. So first minute passed. This is the distribution, pretty even, if I say. And then after 10 minutes, the fly finally learned that I don't want to stay in the middle, right? Okay, the ripe banana probably are ready for food to eat. And then unripe, there's also some fruit flies. So they want you to perform a chi-square test for the 10-minute time point. So that's first. And specify the null hypothesis that you're testing and enter those value in the table below, right here, yeah? So we got to use, basically fill out this table. Okay, so let's take a look at this using that, okay? So let me spread it out again. And this one here, let me do this. Okay, and the chi-square formula is given on the test, so you don't have to memorize it. They're given as part of the equation sheet. And the equation sheet, if you take a look at the equation sheet, is all the way on the top. Okay, so let me show you the equation sheet right here, right? So this is the chi-square table. So really you just need the chi-square table and chi-square formula, yeah? You don't need the standard deviation, just the formula. Alrighty, so this is the data. We got to use the data and the formula to come up with chi-square calculation. Okay, let's do the chi-square and then figure out what the null hypothesis should be. So this is the, do you see the column? The first column is observed, then is expected, and then the third column is essentially your chi-square, right? So this third column allow you to calculate the chi-square. So maybe I'll just write it down here. This has to do the chi-square calculation here. Okay, so what is the expected in those 3 chambers, right? So the way you set up expected depends on your null hypothesis. Your null hypothesis determines the expected result. So this is something we need to memorize. Null hypothesis is not hypothesis. Your hypothesis can be anything, but null hypothesis can only be one thing. Okay, so null hypothesis will essentially, in a very general way to say this, is that what you expect to see. So what is expected, okay, to be seen, yeah? What is expected if there's no factors that influence the fruit fly behavior? So what would be the null hypothesis? So let me write it bigger, yeah? Okay, is what you expected to see if there's no differences in any of the experimental groups, yeah? Gives the expected result and is what you, and gives you what, and is what you expected to see if there are no, if there are no, if there's no deciding factors on what can influence the fly behavior. Yeah. Okay. The fruit fly behavior. Okay. So in that if this is the very easy way to define null hypothesis, so what is the null hypothesis here? Okay, the answer would be that there should be equal number of fruit flies in the three segments of the choice chamber. In the chamber with the ripe banana, in the middle chamber, in the unripe chamber, they all should be the same. Remember, we have 60 fruit flies. We should be seeing 20, 20, 20. So that's how you get the 60 fruit flies. Again, if there's no deciding factor, that means take out the bananas, right? Then what do you expect to see? Then there's no difference between any of the experimental groups. So you're expected to see even distribution. Yeah. So I don't know how to say that properly, but that's how I think about it, that no hypothesis give the expected result, and it's what you expected to see if there's no deciding factors on what can influence your experiment. In this case, the fruit fly behavior. Okay, so and if you put that in words, your null hypothesis, um, is that okay? Yeah. So you kind of have to again, there's different way to say this. I would just say that there's equal. There are equal. Ah, yikes! Give me a sec. There are equal distribution of flies in all three portions of the choice chamber. Yeah, of the chamber. That is my null hypothesis. Okay, but what you actually saw, which is the observed, is from your experiment. And this is what we actually saw, right? We saw that there is a difference and it's based on time. So do I use 1 minute or 10 minutes? Here it tells you. It says to do the 10-minute time point, to do a chi-square for the 10-minute time point, not 1 minute time point. So you got to be careful too. Sometimes the time point does make a difference. So if you look at the observed here, would be 45 for the ripe banana, and 3 for the middle, and 12. And this also add up to 60. Yeah, they have to. Okay. Now you're ready to do your chi-square test. Okay. So if I do this on calculator, you just subtract 45 and 20, doesn't matter, square them and divide by 20. That's what this formula says. You'll get 31.25. And again, this one is 3 minus 20, negative 17 quantity squared, and divide by 20. You're going to get 14.45. And the next one is 12 minus 20 squared divided by 20. You get 3.2. And then chi-square means you have to now sum. You got to add all these together. So do you see the chi-square formula? It means you got to sum all these three data points. So if I add them together, that is my chi-square right here. Yeah, so I just add them. And this will give you 48.9. This is the chi-square, okay? Is 48.9. So we did the chi-square and we specified the null hypothesis and we entered the value, yeah? So this would be 3 points here. One for the null hypothesis that the flies will be evenly distributed. That's the only way to get the expected result, okay? And once again, null hypothesis is how you get the expected, and your actual experiment is how you get the observed, okay? So always, expected is how you know hypothesis. If I flip a coin, my null hypothesis would be half head, half tail. That is the null hypothesis. But hypothesis could be anything. Hypothesis could be, for example, you can say that you're going to get more heads than tails because the coin is rigged, right? That would be a hypothesis, but that's not the null hypothesis. So observed is the result you get from the experiment. So from experiment, you get the observed, and from null hypothesis, you get the expected. Alrighty, so now you got to answer part D. Explain whether your null hypothesis is supported by the chi-square test and justify it. So by looking at the chi-square, I need to now switch to the degree of freedom, right? Your degree of freedom, in this case, you can use either one, but the default is 5%. So use the p-value as 5%. And then which degree of freedom? degree of freedom would be 2 here because it is the number of outcomes minus 1, yeah? So degree of freedom in this case is the number of outcome, which is 3 outcomes, right? Either the middle chamber or ripe or unripe, and you take away 1. So it's going to be 2. So under degree of freedom, 3 minus 1 equals to 2. We end up with 5%. We end up with 5.99. That is the chi-square value. So if the chi-square, if your chi-square is larger than 5.99, you need to reject null hypothesis. Something is wrong. That is the case here. Our chi-square is 48.9. So definitely you have to reject your null hypothesis. Your null hypothesis says that there should be equal distribution. It is not. Your experiment rejected your null hypothesis. So because something else is going on, there is something that causing the fruit fly to distribute unevenly towards the ripened banana. And the reason is the banana, right? So that's why you reject it. If your chi-square is less than 5.99, then you accept your null hypothesis that whether ripe banana or unripe banana, it does not make any difference in terms of the fly distribution, which is not the case here. So how do you justify this then? Your justification, of course, you got to talk about chi-square, right? And you talk about the 5.99, and then you just basically say that your chi-square, 48.9, is greater than the 5.99 with 2 degree of freedom, 0.05% p-value. Therefore, we reject our null hypothesis. That is your justification. So let me type that out, okay? So I would, for part D, okay, we know we reject it, but let's just be precise here, okay? Since our chi-square, okay, you can see this, our chi-square is 48.9 and is greater than 5.99, using a degree of freedom equal to 2 and p equal to 0.05. We reject, okay, our null hypothesis, but you got to take one step further, yeah? Okay, there, the banana, the presence of the banana, BNA, something like that, yeah, does affect fly distribution, yeah? Okay, so that's what our result is showing, okay? All right, so that's that one. So this one would be, let's see how many points. This would be, you got to give yourself, this is part D, right? Part D is how many points? Part D is 2 points, right? So 1 point for your explanation and 1 point for justification. So this would be 2 points, saying something like this, but you got to calculate, well, you got to tell them how you got the degree of freedom. Okay, is that right? Let's move on. Okay, so give yourself 2 points for part D, 1 point or nothing, yeah? I think last one is only 1 point left. So part E is 1 point. Propose a model that describe how environmental cues or stimuli affects the fly behavior in the Troy chamber. Now, whenever they say propose a model, just means to explain, yeah? Explain in terms of a model. Okay, so it's kind of confusing, but this is 1 point here we're going after, and I don't want to see an essay, yeah? So a model for that could be, for example, you can talk about the sensory input. So that's what I'm looking at here, okay? Sensory input, that's the first thing come to my mind. That would be the environmental cue, which in this case is the order molecules from banana. And then that will lead to the integration by the central nervous system. And finally, the motor output. Motor means movement. So the motor output is if the integration tells you there's a favorable source of food, your motor output is movement towards that. If the integration tells you this is nasty, then I'm gonna move away. Is that simple, right? So propose a model. So the model would be this. Okay, this is the model for every stimulus, integration, and response. So the sensory input is basically the stimulus. In this case, would be the odor molecules, and the integration is integration of by the nervous system, right? Fruit fly. Okay. So this is the integration. So this would be the processing by central nervous system. And then your motor output will carry out command or response, okay, as determined by integration, yeah? So when they say propose a model, you could tell them this is the model, okay? This is the model of how flies detect. So it says how environmental cue affect, right? How environmental cues affect flies behavior. So you start with sensory input. Okay, step one is the sensory input. So stimulus or odor molecules are detected. Okay, and this can work for anything. It can be light. In that case, the sensory input would be photons, particles of light, and you detect with your eyes, right? The compound eyes detected. And then integration. So sensory input are processed, right? I'll post this on the Google Classroom, yeah? Are processed by central nervous system, okay? So it decides what to do. And then step three, that the flies will have to carry out a motor output, right? So carry out the command response as determined by the brain, yeah? Central nervous system, up that, okay? So that's it, this is 1 point, so definitely nothing more than this. This is already more than what you need, yeah? Okay, so that would be 10 points. Can you please give yourself out of 10 points and just take your time to add them together and put a total out of 10 for question number 1. Is there any other parts that you want me to, that's unclear on the point? Because we want to add up your points and kind of see how you do, yeah? Okay, so let me know, and I'm gonna move on to the next question. Okay. Everything okay here? All right. All righty, so not too bad, but in this new AP that you're going to be seeing in later on in May, just 3 weeks away, you also have to deal with an experiment. Yeah, it's probably something you never seen before, most likely, and then you just have to take the data and do what you can. Okay, so there's going to be one experiment. All right, this one, let's take a look at this one. So the first thing I see here, you probably have seen this before when we did photosynthesis, right? The y-axis is absorption, and the x-axis is the wavelength. Okay, so if you look at graph 1, whatever this molecule is, it loves to absorb waves that is 550 nanometer. Yeah, it's kind of like greenish. Okay, and if you look at graph 2, this is a different molecule. It does not absorb around 550. The absorption is almost zero, but it absorbs around 650 and about 420 or 30, right? And this is nice enough to give you a color code, right? So what is 550? 550 is about green. 600 is like yellow orange, right? And red is like 700. So this is the Roy G. Biv. And these are molecules that absorb light, which makes them pigments. That's what pigments are. All pigments absorb light. Your skin has pigments. Melanin, sorry, not melanin, is melatonin, yeah? And that absorb light, so you get darker. Chlorophyll which is what this question is talking about, different pigments, they absorb sunlight and they can harvest the light energy from there. So this is an absorption spectrum that indicates the relative amount of light absorbed across a range of wavelengths. The graph represents the absorption spectrum from two species, two organisms. One of them is chlorophyll A, found in green plant. This is the one that we know. The other one is found in, it is called bacterial rhodopsin, found in purple photosynthetic bacteria. So one is in plants and very complex organism. The other one is in bacteria, which is very primitive. So the bacterial rhodopsin is evolved first, has evolved first, right? And chlorophyll evolved later. Okay, so one of them is chlorophyll, the other one is bacterial rhodopsin. Okay, and what is the question? The table shows the range. Okay, great. So the question is identify the pigment. Which one is which? Yeah, and you have to explain and kind of justify your answer. Now, the new APs, they're not going to ask you to explain and justify. It's basically the same thing. So this is the old AP, yeah? So this is a little bit unclear that what does it mean to explain and justify, right? Justify using the data and use the data to justify your explanation, okay? So they kind of work together on the new AP. All right, let's take a look at this. So which pigment is which? What do you guys think? Yeah, we should know plant reflect green, meaning that green is not absorbed. This is why something looks green. Every time something looks green, that means green light is reflected. So the graph on the right side, graph 2, is chlorophyll, okay? Because chlorophyll A, they reflect green. They cannot absorb green, yeah? We can only see reflected light. Okay, so if you look at the moon in the sky, the reason you see moon lit up middle of the night is because sunlight hits the moon's surface, reflects into your eyes. You're able to see that light. But there's a bunch of light that just passing the moon, never hit the moon. You're unable to see any of those. Yeah, you cannot see light that is passing. You can only see light that's reflected into your eyes. So you can only see green here. So because green is around 500, 600 right here, that's your justification, right? And your explanation is that you see the reflected light. So here, chlorophyll plant reflects green. So let's put our thoughts together. Okay, again, explaining justification using the data to explain. So graph 2 is chlorophyll A. Okay, green plant. So we talk about green plant reflect green light, which is we're told the green light. So I'm using the data to justify. Yeah, 495 to 570 nanometers. Okay. Okay. And you can even say they absorb blue, violet, and red, orange. Yeah. Okay. That's even better if you know that. But absorbs and absorbs. So blue, violet, and red, orange. So only graph 2. Only graph 2 reflects green. Okay. Okay, so something like those will be fine. Yeah. Okay. And then okay. Let me just say absorb. Absorbed. Yeah, that's fine. Okay. And then I want to say graph 1 is bacterial rhodopsin, right? Bacterial rhodopsin. So don't forget to talk about this guy. And you know how bacterial rhodopsin, they are what? What color are they? If you look at this one, it says it's purple. Purple, where's the purple one? The purple photosynthetic bacteria. So they they absorb green, they will reflect purple, right? So you can even say that. That's what the data is saying, right? So reflects green and absorbs, um, reflects purple, I'm sorry, purple wavelength and absorbs green. Yeah. Okay. So this would be about 550 nanometers, okay, like that. So that would be enough. Yeah. So graph one and graph two. Okay. So explain and justify. Justify using the data. We can only see. Let me see. I'm gonna say that we are able. We are only able to see reflected light waves. Um, since plants reflect green. Okay. Since green plants reflects green, and graph two must be chlorophyll A. Yeah. Okay. And green plant is very green light around this, absorb blue violet. Okay, good. So like that. Yeah. Okay. So any questions on those? Is that right? Um, so kind of weird, right? Okay. So let's look at the next one. Part B. Part B, we talk about an experiment. We have identical organism containing pigments from graph two. So that would be chlorophyll. So plant, two different plants as the predominant light capturing pigment. Now remember in plants, they have chlorophyll A, they also have chlorophyll B, which absorbs slightly different wavelength, and they also have the yellow, orange carotenoids, right? So there's many pigments. So they're saying mostly chlorophyll, okay? Now you separate them into three groups. The organism in each group are illuminated with light. So you use 650 for the first, 550, and 430. So the question is, the three light sources are equal intensity. So remember, everything has to be constant except the wavelength here. In terms of light, wavelength means color. Intensity means brightness. Yeah. So they have to be the same brightness. And only thing that can change is the wavelength or the color. Yeah. So that would be the fixing the variables other than the wavelength. So predict the relative rate of photosynthesis and justify. So this is easy. You just look at the wave, right? So if you look at the three waves here, so I'm going to actually just copy this portion. Okay. And if you look at them right here, okay. And we also have this graph here, right? So we're using this pigment graph two. So you can now see, based on that, which group is able to absorb most light. The more light absorbed, the more photosynthesis. This is the connection that they want, right? The rate of photosynthesis depends on the amounts of light that can be absorbed. So you see group one, 650 is right here, where it's almost peaking here, right? So 650 is actually pretty good absorption, yeah? Group two, 550 is bad, zero absorption. So the plants in group two is unable to absorb any light that is 550. They're going to starve. They're not going to have photosynthesis, right? And 430 seems to be even better. Look at 430 here is right at the maximum absorption. So I'm going to argue then group one, moderate, right? Group two, nothing. And group three has the maximum rate. Okay, so 650 looks like it's right here. And 430 looks like it's right here. And 550 is zero absorption, right? So predict, we did, and justify. Justification would be the more light absorbed, the more energy that can be turned over for photosynthesis, right? Okay, so what do they want us to do? They want us to use, okay, the rate, right? Okay, so my prediction. So again, it's asked you to predict. So I'm going to say group one is median level, right? Median rate, yeah. And group two is zero, none, no photosynthesis, zero rate, yeah, lowest rate. lowest or zero rate, yeah. And group three here will be the highest rate of photosynthesis, yeah. So our justification, okay, after you made the prediction, now we can do the justification, yeah. How do we justify them, all right? So um, group one is what, 650, right? So 650 nanometer have um, has some absorption of light. Well, I'm gonna say uh, chlorophyll A absorbs a moderate amount of light, or median, moderate. I'm gonna use the same term, okay? Median, instead of using median, I'm gonna say moderate now. So I'm trying to use the same term, right? So absorb moderate amount of 650 nanometers and have moderate, okay. Let me just say that, yeah, okay. And uh, and um, uh, chlorophyll A, so continue one more time. Okay, chlorophyll A cannot absorb light with 550 nanometer, as seen in graph 2. And finally, chlorophyll A absorbs, has a huge, chlorophyll A has a large absorption of light of 430 nanometer, as seen in graph 2. Yeah. Okay. The more, and I will just tie up with this one sentence, right? The key point here is that the more light, okay, light energy to photosynthesis, the more light absorbed, the more light absorption, the greater the rate of photosynthesis, right? So that's why my prediction was the way, yeah? So even though it's kind of easy, but when you try to put them into words, sometimes you find out it's kind of awkward to talk about it. So make sure you know how to do this, yeah? Is that okay? So that's pretty easy, right? So this question, because it's a while ago, I think the new AP test would be more elaborate. This is kind of weird here. It's pretty easy, this one. It's just hard to put into words. Okay, let's look at part C. So part C says, now this bacterial rhodopsin, well, that's graph 1 now. So we're coming back to the bacteria, the photosynthetic, the purple colored, the reflect purple. And they have been found in aquatic organisms whose ancestors existed before plants. That makes sense, even though they're from the same environment. So maybe, maybe they gave rise to plants, right? So there's an evolution here then. So that's why it says, propose a possible evolutionary history. So come up with a scenario for plants, okay, that could have resulted in a predominant photosynthetic system that use some of the colors, that only use some of the colors of visible light. Okay, so I gotta read this a few times, what exactly they want you to propose, okay? So they give you a fact, and I want you to propose this, yeah? So let's take a look at how to put this together, okay? So they're telling you that there's also bacterial rhodopsin, even before plant came about. So they want you to come up with a scenario, okay, that how come today, the photosynthetic system today, only use some of the color? Why don't we use all the color? Why don't we just absorb? Why don't we have a absorption that is absorption of every single colors, right? I think that's what they're trying to say. You can see that this is the modern day. Okay, why don't we have a graph that is peak at every single wavelength? Why don't we have absorption? Okay, that way, right? So give them a scenario. There's no right or wrong answer here because nobody knows, right? Why don't we have this like 700 and 400? Why do we Why do we hate green light? Why do we reflect green? Why do we absorb blue violet more than green? Why is that? Right? So they want you to propose one more time a possible evolutionary history or plan that could have resulted in a predominant photosynthetic system that we see today that use only some of the colors of the visible light. Okay. So why don't we use like infrared? Okay, so remember after 700, you're the infrared region. This is the visible. And what is more energetic than visible is ultraviolet, right? That's this way, yeah? So if you notice that plant do not use ultraviolet, even though ultraviolet has more energy, higher energy, and plant do not use infrared. They only use visible light and not all the visible light. They only use a fraction of the visible light. So why? Propose why. Okay. So because they mentioned this bacteria, then I'm going to say there's a transition, selection, natural selection. So you can see that there is a natural selection. So I think that's why we're given the first sentence. So talk about graph one, talk about this bacterial adoption, talk about this and becomes the other. So I think what the question wants you to is that come up with a scenario in terms of natural selection and explain why you have natural selection going from graph one to graph two. And this is just one point. So you don't have to be super elaborate, right? So this is a natural selection. So before plant came about, this is your pigment. They specialize in what? Blue, not blue, I'm sorry, in green, right? In green region. So these love to absorb green. But now they don't. They hate green. They love blue violet, yeah, or red orange, right? So give them one evolutionary relation. So I'm gonna say that well, blue violet provides more energy than green. This makes photosynthesis more efficient by utilizing light that has more energy. So that's one thing that popped to mind. Okay. So, but there can be other things, yeah. So I'm gonna say that uh, okay, pigment, or they use photosynthetic system. Okay, that's what they use. Yeah, photosynthetic system that that absorbs light with more energy. So I want to make sure they understand what that means, right? So 430 nanometer instead of, okay, 400 in graph two, instead of 550 in graph one, yeah, are selected for, are more efficient, yeah, in photosynthesis, are more efficient in photosynthesis. Okay, these systems are selected for, and okay, photosynthesis. Yeah. So these systems are selected for, okay, in plant evolution, right? In the evolution of plants. Yeah. Okay. So that's basically the reason. Is there any other things that you guys see? Yeah. We are proposing a mechanism. Yeah. So there's more. I'm gonna show you some other possible scenarios. Okay. So can we say that, for example, this? Okay. So these are some other alternative answers that you can talk about. So right here. So you could say, okay, uh, that again, there's so many things that you can say here, right? So you could say that, for example, this one, if you absorb light with longer wavelength, it's not efficient in energy, right? And you can also say natural selection favor organism that rely on pigment that absorb available wavelength of light. Okay, endosymbiosis. Yeah, it depends what you say. These are like kind of like scoring guide. They're kind of scattered everywhere. Okay, so you could also say that, okay, it's a mutation. That would be possible. That is a good one. They say the mutation that alter the pigment used by plants. So the pigment no longer absorb green and now it specializes in the other two ends of the spectrum. So that would be a good one. Again, you don't have to give them all possible, right? They're just one particular scenario. So you can say mutation occurred. Okay, that's possible, right? There's no right or wrong answer in bacterial rhodopsin because they tell you that again. Okay, bacterial rhodopsin that altered the absorption used by the organisms, and now lights, and now instead of absorbing green light, uh, blue, violet, and red, orange lights are utilized. Okay, so there's no right or wrong answer. Yeah, so this is just give you some idea. You can blame on everything. Okay, let's just look at one more. Remember endosymbiosis? That's when chloroplasts were eaten. So what they're saying is that endosymbiosis happened, and instead of eating bacterial rhodopsin, they're eating another bacteria that use blue, violet, and red, orange. Okay, so that means that okay, so now the present day green plant do not use bacterial rhodopsin because they weren't phagocytosed to become the present day chloroplast. See, that's another kind of BS, right? So I call them BS because you're just coming up with whatever makes sense. You just need to have one particular scenario in the multiverse, yeah, of evolution. Again, propose one possible evolutionary history that could have resulted in present day photosynthetic system that use only some of the color. Okay, so I use natural selection, and the answer here could use mutation. You can also say endosymbiosis that ate up cyanobacteria that use blue, violet, and red, orange instead of uh, bacterial rhodopsin. All right, so that's the points here. Now the point system is not so clear here, but let's try to put the points in this. So part A, okay, so let me write down the points so we can add up the points, okay? So part A is how many points did we say? Part A is 3 points, right? We have 3 bold terms, so 3 points. So roughly give yourself, if you feel like you fully answer the question, please give yourself 3 points. And part B, we have 3 systems, 3 system here. So that would be 5 points, yeah? They give you 5 points for some reason, yeah? So 1 point for your predictions, okay? And 1 point for 2 point for justification. I don't know why they give you 5 points only. Yeah, it's annoying. They give you 5 points here, okay? And part C is 2 point left. This is 10-point question. So propose a possible evolutionary history. So I guess you got to give them a reason, right, for your proposal. Yeah. Okay. So I know it's very hard to estimate point, but just do your best. Can you give yourself out of 10 points, please? Let's move on to the next one. Okay. Please do that out of 10 points for question number 2. Yeah. Okay. All right. Next one here, number 3. We talk about fossils. Okay. Let's take a look at that. So fossils or lob-finned fishes are ancestor of amphibians. Yeah, lob-finned, their fins are so thick and bony, feels like their legs. Yeah, and if you can develop or evolve lungs, then you got yourself amphibian. So those are the fish that actually gave rise to amphibians, right? So they're saying that these lob-finned fish, which are like the transitions between fish and amphibian, are found in rocks about 380 million years ago. Okay, fossils of the oldest amphibian-like with true legs and lungs are found in rocks that is younger, right? Because the early organisms are deeper. Okay, they are deeper buried, The younger organisms are higher on the fossil records. So 3 samples of rocks are available that might contain fossils of a transition species between larvae and amphibian. So one is here, one is here, one is here. Okay. Select the most appropriate sample rock in which to search for transitional species between larvae and amphibian. Justify. Okay. So that, I think that's pretty easy. You just have to find a time period between, but you got to be careful because we don't want to miss the time period. So let me just kind of put that into some diagram. Okay. So this is the bottom. So they said this line is 380 million years ago. Okay, so that would be, this is, let's say this is 390 million years ago. So we have fossil here. Okay. And then I also have 300 and, okay, sorry. Let me do this one right here. Yeah. Um, what is the, give me a sec. Let me take this. Okay. And I'm gonna erase this first. Okay. So we have, let me erase this part, not 390. Ugh, what did I do? Okay. So 380 million years ago, right here. 380 MYA. And then amphibians are found 360 million years ago, 363, right? So 363 million years ago. So this is where you find the amphibians. And this is where you find, what is a lob fin. Okay, so if you want to find out what is that, the transitional fossils, so which one would you find? Wouldn't you have to find somewhere in between them? So that's what I meant. So somewhere between 363 and 380 is when larvae evolved, right, into here. So the one that will work is 370. If you look into 350, you're going to find what? You're going to find amphibians only, right? Mostly amphibian, right? So that's where amphibian is. If you find 390, that's too deep. You're not going to find transition because they haven't evolved yet. You're going to find lob fin. So your justification is that 370 million years is between, so it depends how you say it, right? So let's put them in words now. Okay, so always putting in words is a little bit tricky, yeah? So the answer is 370, you get 1 point. And these are 4 points question now, okay? So your justification then, how do you justify this, guys? You have to put this in, right? So it has to be before the emergence of amphibian and after the what is this the larvae, right? So 360 million years ago, a layer is okay, is before, okay, the the oldest, I'm using the same wording, okay, as the oldest amphibian like amphibian like animal, and is before and before the before the what is this the rocks are at least 280 before the last lobbed finned fishes. Yeah, so that would be my justification. Yeah, okay, so the transitional fossils are found between them. Between what? Between the oldest amphibian, so the first one to evolve. That's what, before the oldest or the first amphibians. Yeah, that'll be the oldest, right? And the latest or the last will be the latest. Yeah, okay, latest or last. lobster fish. Yeah. Okay. So that kind of gives you a sense, putting them into words. You just have to, it's playing with timelines, kind of weird. All right. So that would be the justification. That would be 2 points. One for 370 million years ago, and one for saying that transitional fossil. Yeah. So transitional fossils must be found uh, in layers uh, right here before the oldest amphibian-like animal, and the latest lobster fish. Okay, all right. Now, second part, describe two pieces of evidence. That's the 2 more points. So 1 point for each evidence. And when they say describe, just means write about it. What is the characteristics? Yeah. What is it? Yeah. What is it used for? What is the purpose? Whatever description you can, just don't state it. Yeah. Stating it is just one word or something. Yeah. You want to kind of describe what you're stating. So two pieces of evidence by fossil of a transitional species that would support a hypothesis that amphibians are evolved from lobster, right? So what two pieces of evidence? Now, fossils provide morphological evidence. Do you agree? Okay, the way they look. That's why we look at fossils. So fossil give you a very convincing evidence of evolution because they tell you exactly what the organisms look like, their forms, their structures, the arms, the legs, what kind of body parts they have, right? Okay. They could also give you molecular evidence, depending how old the fossils are. You might be able to find some evidences of molecular biology. So out of all the evidence of evolution, you want to focus on, in this case, is the morphological data. Okay, so this is what I meant by description. If you simply put down morphological data, that is not a description. So then you start to do such as what? Such as structures, right? Limbs, okay, or legs, okay. You can also talk about um, um, lungs, right? Respiratory structure, lung cages, rib cages, maybe. Because talk about lungs, soft tissues, they don't fossilize. So talking about legs, arms, rib cages, okay, that, okay. And also, I'm gonna say molecular DNA data, okay, that can provide, sequence homology of the transitional sequence similarity of the transitional species, or that can provide sequence similarity that links the transitional species to amphibians. Yeah. Okay. So that would be a description. So instead of just writing down a statement, what these are, like DNA, you just talk about how they can do that. If the DNA sequence that can link fish that can link transitional species to amphibian, that's fine. Yeah. All right. So can you give yourself points for this? You can also talk about teeth. You can talk about uh, um, you can talk about the vertebrae here. You can even talk about, do they live in the same area? So, okay, but I usually focus on, in terms of evidence of evolution, I always look at the fossil record, biogeography, the distribution of plants and animal, comparative anatomy, right, which is the morphological evidence. You can look at vestigial structures. You can look at DNA, RNA evidences, right? So those are all evidence of evolution, right? So they want you to apply them to this particular example. So 4 points. Please give yourself out of 4 for this one. Yeah. Let's move on to the next one. Okay. Next one is this one. Okay, so matter is recycled continuously. This looks like one of those 4 cycles. We have water cycle, we have Kelvin cycle, we have nitrogen cycle, and we have phosphorus cycle. So this is carbon cycle. How do I know? Because CO2. The only cycle that has carbon is carbon cycle, has CO2. Yeah. So if you look at CO2, somehow on the diagram, CO2 gets incorporated into organic molecule. Okay, so that must be photosynthesis. Right away, you should be able to write down and make sure it makes sense that process one is photosynthesis. It converts CO2 from air into organic molecules in plants as glucose. This organic molecule can be glucose, can be starch, right? Basically, that's how plant gets all the organic matter. Even lipids come from photosynthesis. Photosynthesis produce not just sugar, but a bunch of lipids for plants as well. And process two returns them back as CO2. So process two must be cellular or aerobic respiration. So either one is fine, aerobic respiration or cellular respiration. And that's how plants returns them, right? And cell respiration always come up with mitochondria, and photosynthesis, of course, will be chloroplast. All right. So right away you understand this, right? So matter is cycle, carbon is cycle. Now part A says identify a key metabolic process for step one. We did, and for step two, we did. Explain how each process promotes the movement of carbon. For each process, your explanation should focus on the role of energy and the movement of carbon. Alrighty, so the identification is not hard, but now we have to make sure when they ask you to explain, you are using the terms and the concepts that they want to see in your explanation. So we're going to talk about each process. So you cannot just talk about photosynthesis. And what do you talk about? And here specifically says, talk about the movement of carbon and focus on role of energy in the movement of carbon. So movement of carbon and role of energy. Okay, let's give it a try. Okay, so I'm going to type it out for each process. I'm going to start with process one. So starting with photosynthesis, what can we talk about? Photosynthesis, okay? So if you recall, every time you think about photosynthesis, you should think about light and dark reaction, yeah? Okay, so very, very quick review for photosynthesis, and which is essentially what we need here for this question is you can think about photosynthesis as having two boxes. The first box is the light reaction. They depend on light. Okay, the second box is the dark reaction, which is also called Kelvin cycle. Okay, so I'm going to draw a circle here. This is called Kelvin cycle. It is also called dark reaction. We need to know what goes into each step and what comes out of it. Light reaction is the electron transport chain. Okay, this is the chemiosmosis. So if you have electron transport chain, chemiosmosis, you are going to get ATP. So ATP comes out of it. And also the electron-rich energy carrier, NADPH, comes out of it. Water goes in, and water is split and is a source of oxygen. Water is the electron donor. It donates electron to the electron transport chain, and water becomes oxygen, which is a byproduct. It does not continue. Oxygen is released out of the leaves for everybody to use. Now, also light have to go in, okay? So this is your light reaction. Three things goes in, one thing go in, water and sunlight, right? Okay, water goes in. And three things comes out, but only ATP, NADPH continue. So if you look at ATP, ATP will enter dark reaction, NADPH also enter dark reaction. And also one more thing enters would be the carbon dioxide from the air. This is how carbon is fixed. And what comes out is a 3-carbon sugar called G3P, glucose. This is glyceraldehyde 3-phosphate. And this G3P is synthesized into glucose. It can be turned into starch, or it can be turned into lipids and other biomolecules. So when they talk about the movement of carbon, guys, do I talk about light reaction or do I talk about dark reaction? Then you need to focus on Kelvin cycle because carbon is fixed by photosynthesis. So you want to talk about at least Kelvin cycle, right? That's when carbon dioxide is fixed. And when they focus on energy, then what are energy here? ATP is energy. Why do we need ATP? ATP powers Calvin cycle. NADPH is energy. It carries high energy electrons to reduce carbon dioxide into sugar, right? That is the energy, and they come from the light reaction, ultimately sunlight. So that would be the strategy here moving forward to talk about that. So let me write down. There are three things that Calvin cycle needs. ATP. This provides energy to power Calvin cycle, because Calvin cycle needs to fix carbon into sugar. It takes energy. And second thing, okay, second thing is NADPH. So this provides high energy electrons. So the energy rich electrons to reduce CO2 into glucose. And third thing, CO2 provides carbons, provides source of carbon in glucose. So you need to fix CO2. That's why Calvin cycle needs three things. So if I were to talk about photosynthesis now, I need to focus on the movement of carbon and the role of energy. So process one, okay, the Calvin cycle. So I'm gonna say Calvin cycle fixes CO2 from air, okay, which come in from leaves, yeah. So CO2 enters the plant from leaves. So what about them? This is the movement of CO2, okay? Fixes CO2 from air. So I'm gonna say from air, okay, via leaves, yeah, okay, into 6 carbon sugar, yeah, glucose. There, that's the movement for process one, okay. Calvin cycle uses energy from light reaction. That's the energy, yeah, in the form of, okay, it receives ATP and NADPH, right? It receives ATP. Let me see. Calvin cycle, how about just directly? Calvin cycle receives ATP and NADPH from light reaction. Okay, ATP powers reactions in Calvin cycle. Okay, ATP hydrolysis, right? And NADPH provides high energy electrons to reduce CO2, to make glucose. High energy electron to make glucose. Okay, so that's it. So we answer both of the parts. All right, now we're going to talk about cellular respiration. Yeah. And whenever you think about cell respiration, you should think about glycolysis, Krebs cycle, and electron transport chain, which is chemiosmosis, right? So if we do also a very quick review, yeah, we do need to know this, but I think if they want very much detail, they will give you the process. But okay, so we need to know this, that when you talk about cell respiration, okay, let me draw this out, please. Give me a sec. Okay, so this would be, where's my pen? Okay, right here. Okay, so I'm gonna use blue. So actually, I'm gonna write it. I'm gonna type it out. Okay, so three main steps for cell respiration. Yeah. So glycolysis, I need you to memorize, is that it is universal. Whether you go through aerobic or not, you have to go through glycolysis. It is, these are the terms associated with glycolysis. Universal in all living organisms, they all use it. So it's present in the earliest common ancestor of all living things. That's how you say it, yeah? It's present in the earliest common ancestor. Okay, so this process of making energy present in the earliest common ancestor. How do I say it? Spell earliest, okay, common ancestor of all living things, bacteria alike, yeah? So that's why it's called universal, okay? So make sure we understand what universal means when we talk about evolution, yeah? It is also anaerobic, okay? So what is that? What is the significance of anaerobic? It is anaerobic means that the original Earth had no oxygen. Okay, so glycolysis was that early. They had to depend on glycolysis. So living things then depend depend on glycolysis to generate energy. Yeah, for energy. Okay, and it's also the common pathway. So common pathway between what? Between aerobic and anaerobic. Anaerobic means fermentation. Yeah, and aerobic, which is cellular respiration. Yeah, respiration. Okay. So whether you go through alcohol or lactic acid fermentation, you got to go through glycolysis. And the fact that glycolysis are in cytosol suggests that this came before the evolution of eukaryotes. Okay, glycolysis evolved early before eukaryotes were around. Chaotic cells. Okay, so these are the thing about glycolysis. Anyway, I digress. Yeah. So glycolysis, and then you have Krebs cycle. Okay, so let me write it again. Number one, glycolysis, Krebs cycle, and then you have electron transport chain. But I want you to think about electron transport chain, and every time you talk about electron transport chain, you got to talk about chemiosmosis. Yeah, osmosis. So they are talked about together. So electron transport chain harvests energy from electrons, and chemiosmosis uses those energy. Now it's in the form of proton gradient and ATP synthase to generate ATP. Okay, so what is the purpose for glycolysis and Krebs cycle? They actually oxidize glucose. I want you to think about that way. So 1 and 2, glycolysis and Krebs cycle, oxidize glucose completely for its electrons. Glucose has high energy electrons, okay? So they oxidize glucose for high energy electrons. You need electrons, okay? And these electrons are stored in NADH and FADH2s. And these electron carriers will carry them to electron transport chain, yeah? So these NADH, FADH2s are electron carriers we use for cell respiration. They carry electron with lots of energy to electron transport chain inside the mitochondria. So what is electron transport chain? Why is it necessary, right? It turns out that the only way to harvest the energy we have stored as electrons is to run them through electron transport chain. So the only way we can, living things, not just us, yeah, living things can tap, living things can extract energy from electrons is to run redox reactions on electron transport chain, yeah, redox reactions on ETC. And the energy released and harvested from electron transport chain is used to pump protons, creating proton gradient. So that's the next part. The energy released by electrons on ETC, yeah, on electron transport chain, is used to pump H pluses to create a proton gradient, which is a form of energy itself that ATP synthase, that magic enzyme, yeah, uses to make ATP via biochemiosmosis, yeah. So that's the minimum you need to know, yeah, for electron for the cell respiration. So where is the movement of carbon? So carbon from food eaten, broken down by in the form of glucose, broken down by glycolysis and Krebs cycle. That's it. And then energy, the role of energy in this movement. So energy is extracted by electron transport chain and turned into ATP. So that's what I would put for here. So let me summarize that. So cell respiration, carbon is obtained from food, right? Okay, in the form of glucose. Okay, glycolysis and Krebs cycle completely oxidized glucose into CO2. So you got to talk about, because we're talking about the movement, right? Into CO2 now, into CO2 molecules, okay? The energy from glucose, the energy from uh, glucose in the form of electrons is extracted by electron transport chain and ultimately uh, turned into ATP and ultimately stored as ATP. Energy is stored, right? Not turned into, right? Stored as ATP by chemiosmosis, yeah? Osmosis. So in this case, now we addressed what they want, the movement of carbon we did, right? From glucose broken down by glycolysis, Krebs cycle into carbon dioxide out of our mouth, out of our lungs. And the role of energy, because these movement allows you to oxidize glucose for electrons, we process them, we synthesize ATP from those energy. All right, so you may not be as detailed as this, but something along those lines, that would be great. Yeah. Okay, so that would be 1, 2, 3 points here. So 1 point for identifying both processes and 2 points for explaining process 1 and 2. Yeah. Okay. Could you give yourself 3 points, 2 points, or no point for part A? And part B is simple. Identifying an organism that carry out both. You cannot say any animal. Animal can only carry out cell respiration. So any kind of plants would be fine. Tree, okay, or any plants. Or you can say algae, green algae. So it doesn't matter. Just put one. Or you can say photosynthetic bacteria. Okay, cyanobacteria is photosynthetic. Yeah. So any one of those is fine. You don't have to list all of them, just one organism. So 1 point for identification. You don't have to explain, just identify. So please give yourself 4 points for this question. Okay, let's move on. Next one here. Okay, so any questions so far? We're using these tests to review photosynthesis and cell respiration. So please, you gotta memorize this. This 2 flowchart has so much information that you can talk about. Okay, all right, let's look at this one. So this one here is question number 5. So this is still 4 point question, yeah? Okay, so this is still 4 points. Okay, so let me write that down. And we'll take a break a little bit, but let's hang here. We're almost there. Yeah, 4 points. Okay. So if you look at this question here, is talking about, I would say this is called molecular biology, evidence of evolution. You can look at the amino acid and tell which of the 4 species are more closely related to each other. But upon second look at this, I'm actually looking at the mutation. Yeah. Okay. So let's take a minute on this. So the table below shows the amino acid sequence of a conserved polypeptide. Now, when they say conserved, that means the polypeptide in general didn't change all that much between different species. So it's highly conserved. That means very little changes. So this is quite conserved, but I can see that it is still different. If you look at species 1 and 2, this is the difference right here. I have a position number 4. Species 1 is a valine, but species 2 is a lysine, right? And if you look at species 3, we're comparing to species 1, okay? That's our reference. So species 3, the difference is, you can see, is that it's missing 2. What the heck? Right? And species 4, okay, so let me use a different color. So species 3 is missing 2 relative to 1. 1 is like your control. You're comparing everything to 1, okay? And look at the last guy. The last guy here, there's a bunch of changes. Starting with amino acid number 5, everything is different, okay? So they're different, but yet they're highly conserved. And an amino acid can be like 300, a protein can be 300 amino acids long. We're looking only at the 10 of them, yeah? So we're not looking at the entire protein, just a fragment of the protein, okay? All righty. So, so here's a question, part A. So assuming species one is the ancestor, so that means 2, 3, 4 kind of diverged and then they evolved into 2, 3, and 4, maybe. Explain the most likely genetic change produced in the polypeptide in species 2 and the genetic change in species 3. Okay, so when we talk about mutations, there's point mutation. That is the most common discussed mutation. Point mutations on AP means that change at nucleotide level. Nucleotide level, okay? Not chromosome level, yeah? Chromosome level means that you can break a chromosome, you can lose a piece of the chromosome, right? You can have an extra piece of chromosome. So we're not talking about that. We're talking about the TAGC nucleotide level. So there are substitution, there are insertion, deletion, right? Deletion. So you lose a nucleotide, or you can lose 2 nucleotides. You can also have insertion. Yeah. Okay. So a good substitution, if you look at the substitution, there's usually 3 consequences. And you don't need to memorize the codon table, right? So let me draw a codon table here. You don't memorize the codon table. If they want you to use codon table, they will give it to you. But you should remember, codons are on mRNA. So we're looking at the mRNA, like a ribosome would. Ribosome also look at the mRNA, right? So if you have a substitution, there's 3 possible consequences of a substitution. For example, if you look at the top left, UUC and UUU, they're the same amino acid. That's one consequence. Even though there's a substitution, you still code for the same amino acid. So that's the first consequence, yeah? So if you have a substitution here, you could have a silent mutation. If it's silent, that means that even though there is a change in nucleotide from UUU to UUC, you still code for the same amino acid. So the phenotype never changed. So it's the same protein, same phenotype. The expression of the gene is identical. But you can also have a missense mutation. That means, indeed, the amino acid changed. So for example, UUU becomes UUA. That would, if you look at the codon table, right, change from PHE, which is phenylalanine, to LEU, which is leucine. So the amino acid actually changed. And that can be okay for the protein. That can also be deleterious for the protein. If this change were to happen in the middle of an active site, then it might change the shape of the active site that renders the protein non-functional, loss function mutation. If this happens somewhere else away from the active site, then maybe the protein still have identical function. So we don't know, okay? And you can talk about either one, it doesn't really matter. And the final one is leading to a stop codon. So that would be a nonsense mutation. So in this example, if you look at the stop codon, I can have a UAC, which is tyrosine TYR, becomes a UAA. That triggers a stop codon. So let me use that example, okay? So UAC is TYR, stands for tyrosine. If you have a mutation, a substitution give you UAA, well, that becomes a stop codon. And if that were to happen, you get a shorter protein. The protein have to stop translation before it's completely done. So because of that, you can see that species 2 is a substitution that resulting a missense mutation. Okay, so let me write that down. So species 2 had a new genetic change is a substitution, yeah? Okay, so let me stretch it out a little bit. So this is part A. I have to explain it, right? So species 2 had a, I'm gonna say a substitution, point mutation. That point mutation that resulted in uh, amino acid, a valine resulted in valine to lysine substitution. Yeah, substitution. Or you can, the first substitution, just say that it has a had a point mutation that resulted in a valine to lysine substitution. Yeah, that's probably better. Okay, so explain the most likely change, right? Okay, so and then species three uh, had a uh, had a point mutation that resulted a stop codon in starting in amino acid 9. Yeah, amino acid at position 9. Yeah, at position 9. Okay, uh, so that's it. Okay, so this caused protein to protein this caused premature translation, right? Okay, termination of translation. Yeah, and a shorter protein. Yeah, so make sure you say something like the protein is shorter. That's how we know. Okay, now what about number 3? Yeah, number 4. Number 4 is a friendship mutation, and you get friendship mutation from deletion and insertion. So deletion, insertion, you end up with reading frame changed, and friendship mutation can have any combinations of the 3 above. It can be silent for some of them, it can be missense, and it can also cause a termination early, right? So friendship mutation is really bad. So from the point of friendship right here, there must be some kind of nucleotide insertion somewhere in amino acid number 5 that shifted the reading frame 1 unit down or 2 unit down. Either from a deletion or insertion, doesn't matter. You're going to change the reading frame. And that causes every single amino acid expression to change. Okay, so that will be part B. So part B, predict the effect of mutation due to 4 justify. Oh, okay, so not part B. So part species 4 is a friendship, but that's not what they want you to focus on. They want you to focus on what is the effect of that. Okay, well, that will change the protein 3-dimensional structure because the primary structure has altered, right? And that can cause a protein to be no longer functional. Without the proper 3D structure, the protein has no function. The expression doesn't work anymore. The protein has no more function or less than function, right? Okay, so I'm gonna talk about that. So species 4 underwent or had a friendship mutation, yeah? Okay, underwent frame shift mutation. Okay, so now the consequence, yeah. So the effect of the mutation, right? Okay, so this changed. Okay, this uh, this shifted the codon reading frame, starting at position 5. Okay, um, okay. As a result, the final protein, so talk about the primary and tertiary structure and function, yeah, have different primary structure and likely different overall tertiary structure, yeah. Okay, and now you got to talk about the function, right? So when the proteins tertiary structure or 3D structure is changed by friendship mutation. Okay. The shape is different and will likely will lose its the shape is different. Okay. Um, and will the function is lost, right? Okay, so how do I say that? As a result, the final protein have different primary structure and likely a different overall tertiary structure. When a 3D shape is changed by friendship mutation, okay, its function is lost. Okay, like that. So the final prediction on the structure and the function, structure changed, the three-dimensional structure or shape is changed, so the function is lost. And that's my prediction, right? My justification is because there's a reading frame, okay? So the reading frame. As a result of so reading frame, okay? Reading frame changed. That shifted the reading frame. So all the amino acids starting at number 5 change. So this addresses both parts, okay? So you can read it several times. They want you to predict the structure and the function, which we did. The structure changed because it's tertiary structure, and the function is lost, okay? And our justification is the frame shift mutation. So can you please give yourself points here? So it's 2 points for 2 points for each part. Yeah, 2 points for part A. Part A talk about species 2 and species 3, right? So make sure you explain it correctly. And 2 points for part B. One is your prediction, and your justification has to evolve around frame shift, the reading frame changed. And if you can tell them it started at position 5, that would be great. So everything from position 5 and forward is going to change. Yeah. All right. So that's number 5. Okay. Now 6, 7, 8 on the old test are 3 points each. So let's take a look at those. So these are 3 points questions. Okay. So can you give yourself out of 4 points now for question number 5? And we're looking at a 3-point question. Okay. So this one is identifying the cell parts. Okay. Give me a sec. Yeah, we'll identify. So we'll take a break in 10 minutes. Yeah, about 9 minutes. Now, give me a sec. So here you have this one here. We have cell X, cell Y, cell Z. Just identify what they are. One, and explain how the data supports the classification. Now, there's multiple answers here. Okay, so the key point here is that first of all, they are eukaryotic cells. So if your answer says they are bacteria, no, right? Okay, they're not bacteria. So you cannot talk about bacteria when they tell you all 3 are eukaryotic cells. All right. So they want you to know the function of the organelle. First of all, what organelles have lots of mitochondria? It must be something that's very active. So that pops to mind. Okay. So what kind of cells use a lot of mitochondria? Cellular respiration must be some cell that's very active. So they want you to identify a function. Now remember, the key word here is they want you to identify what is the job for cell X. You don't need to give them an example. Okay, you don't have to say it's the muscle cell. That is not right. Even though it did answer the question, is what is the function of muscle cell? So for movement, for transport, right? So that would be the function. But here, they don't want you to identify the type of cell. They want you to identify the function of the cell. So don't just say muscle, right? Muscle, you get no point. But you have to say, what is the function of cell X? So you can put down for part A, okay? Because cell X can be any muscle, yeah? Any cells. Cell X function is, okay? So what do you guys think? For movement, for transport, anything else? Or transport, any one of those. You don't have to say both, yeah? For, yeah, movement and transport, to pump stuff, yeah? To pump ions, okay? So any of those can be fine, yeah? Do you guys have any other ones? Okay, now we gotta look at the data. So the data I'm using here is mitochondria. It has cell X has lots of mitochondria um, needed to power its large activity, right? Or energy requirement, yeah? So energy requirement. So energy requirement, so usually to move, to pump stuff, to do active transport, okay? So you can say anything along those lines. So there's more answer than what I have here, okay? So is that all right? Okay. And what about cell Y? So the key thing for cell Y that pop out is rough ER and smooth ER. Whoa. So we need to know the function of rough ER and smooth ER. You can even talk about Golgi. This is designed to transport stuff because the entire rough ER and Golgi is to transport stuff out of the cell into the cell, transport protein. So again, the function is to excrete, to manufacture protein, to, this can be a liver cell that detoxifies stuff. That's what smooth ER does, right? So you could talk about it's for secretion, the function, okay? Function is to secrete protein products, okay? To manufacture proteins, okay? So the evidence is lots of rough ER and Golgi, right? Golgi ships the protein to release it out of the cell to incorporate into the membrane or to other parts of the cell. And that's what that is. You can also say cell Y function is to, because of smooth ER, lipid synthesis. That's what smooth ER does. Detoxification or store calcium ions. Okay, so this is the function of smooth ER. Lots of smooth ER, and smooth ER does these three things. So you're just identify the function, okay? All right. All right. What about Z? Z is weird because there's nothing. No. There are none of these. And then some people say, okay, that must be a bacteria. Once again, these are eukaryotic cells. Okay. So there are cells in your body, two types of cells. For example, red blood cells has nothing. Also, platelets have nothing. Okay. But we're not identifying the type of cell. I'm just saying, yeah. So red blood cell and platelets, there's no nucleus and organelle. So platelets is to clot something. When you have a cut, that's what platelets are for clotting. And red blood cells to transport oxygen. So they don't have anything. So these cells have no organelle, no nucleus, yeah? No nucleus, no organelles. Okay, they want to maximize the cell space to transport whatever they're doing. So how do we answer this? Because cell type Z seems like is to do exactly that. It has nothing. So you would say the function is to transport, yeah? And absence of organelle maximize oxygen transport. If you think about Z as a red blood cell, okay? So cell Z function is to transport oxygen, for example, and absence of organelles allow maximum O2 storage, right? Storage and transport, okay? You can also say that you can even say this is a dead cell and no function at all. Okay, so there's so much stuff. I'm just gonna give you some of the scoring guideline that you could talk about for cell Z here. And you can talk about it's like a skin cell or something, nail or cork. Cork is plant skin, the skin of the tree. So they don't really have much, yeah. Okay, again, you don't have to name the cell. Okay, so that's that one. So 3 points, one for X, Y, Z, please. Yeah. Okay, all right, so almost there. And then this one here. So let's finish grading this one and we'll take a quick break. Yeah. So this one, number 7. Number 7 is pretty easy. They just looking for one scientific question that you could use to investigate this. Seems like if you drink water, you pee 3.5 mil. If you drink 1% alcohol, you pee 3.8. If you drink 5% alcohol, you pee more. So this shows you that alcohol makes you pee. Yeah. Okay. So one scientific question that you're investigating: does increasing alcohol consumption causes more urine output? That's exactly what I'm seeing here. Okay. Does increasing alcohol consumption cause more urine output? Okay. So there might be some other question, but scientific question means that there has to be able to do experiment on. Like if I were to do experiment, I can do this. You cannot talk about philosophical question like, do alcohol cause more uh, miscarriage? No, not miscarriage. More divorces. Yeah. Well, that wouldn't even make sense. You can't really test a moral question, philosophical question. Is alcohol good for you? You don't do that, right? So scientific question, that's testable. Now, state of hypothesis that could be tested. So your hypothesis is that whatever you want here is not a null hypothesis, right? Okay. So I think A and B are very close together, right? But hypothesis, if and then, that's a common way to do a hypothesis. You don't have to do hypothesis, right? But I would say that if rat drinks alcohol, higher percentage of alcohol, okay, then uh, um, rat loses more water in urine. So that would be, for example, a hypothesis, right? So A and B are really alcohol. A and B are really close. So a scientific question can be anything, okay? And even similar to your hypothesis. This hypothesis is talking about rat and you're collecting the urine and changing alcohol. So you can talk about this, right? Okay. And then, so last one, using the data, describe the effect of alcohol. Oh, so this is like repeating over and over again. So these kind of questions are not going to be very important on AP. It's going to be a lot harder. So effective alcohol, ethyl alcohol increase urine output. Feel like I'm talking about the same thing 3 times. Yeah. Increase urine output. Okay, that's it. So 3 easy point. Even if it's kind of repetitive, it's okay. So the question is your hypothesis. Now the last one, very quickly here. This looking here, you're looking at a signal transduction. The 3 steps of the famous signal transduction. That's how cells communicate with each other. So this is very important, right? So signal transduction means that the cell use signal transduction to communicate, like nervous system use signal transduction. So this has to do with cell communication, yeah, via signal transduction. And that's usually 3 steps. So step 1, it would be the receptor binding. Okay, so this means the ligand binds to membrane receptor. Okay, now what this will do is that it will trigger the receptor to turn on some enzyme. Yeah, it's kind of weird. So literally, okay, sorry, I'm a little bit lagging here. So this is the receptor binding. Okay, the second step is this. So as a result of the binding, oh yikes, I don't know what happened to my. So here we go. Okay, it doesn't like me to type more words. Okay, so here, so step 1. Now step 2. Okay, so step 2 here. So step 2 is the cascade. So the reaction cascade, a cascade of reaction. How about that? Okay, a cascade of reaction is triggered, is activated, basically. Okay, often using a secondary messenger. A secondary messenger. Now, the purpose for secondary messenger is that it amplifies the original signal molecule. So if you have 10 original signal molecule, you can produce 20,000 second messenger, and each one of the second messenger can help you to activate reactions to amplify the original primary messenger. Okay. And then step 3, you can see, is the cellular response. In this case, the response is to turn on some genes. That's why it is entering the nucleus. Not the messenger itself, but some molecule goes into nucleus. So you can turn on gene expression, which means that you basically turn on translation, transcription, translation. Yeah, transcription and translation of a gene product. Okay, and the gene product can be an enzyme that is critical, that is required, that is the response to this primary messenger. Yeah, in the very beginning. Okay, so this is the most important part of cell communication. This can apply to how hormone works in our body. So this one wants you to explain the role of each step. So 1 point for each step. The role, right? The role for number 1 is to trigger receptor binding triggers step 2, yeah. Initiate a cell contact, yeah. And step 2 is to amplify, to turn on a cascade reaction. And step 3 is the cellular response. In this case, turn on the gene expression. So a protein is made. The protein can be a glucose transporter that allows the cell to upload glucose. All right, so give yourself 3 points here. So before we go to break, can you add up all the points, 1 through 8? It's going to be out of 41. And if you can give yourself out of 41 for free response, that would be great. And then we'll look at the multiple choice, the ones that most people miss. So give yourself out of 41, and then we'll help you convert to AP score later. Yeah. So let's take a 5 minutes. Yeah. And just walk around a little bit. I'm going to mute, and then I'll be back about 5 minutes and start yelling. Yeah. Okay. All right. Okay. All right, guys. Hi. Are you guys? Can you guys hear me? Yeah, Sunny, maybe. Yeah. Hi. Okay. So let's get started because we're gonna look over multiple choice. Multiple choice is pretty easy, so I thought I'd spend a little bit more time on free response. So let's take a look at multiple choice. We're probably gonna skip around so that we focus on more difficult questions that everybody miss, and then you feel free to let me know which one you want to go over as well. Yeah. So I think Office is sending me essentially the question that most people miss. So that way we can just go over those, yeah, for now. Okay, so first is I want to look at the data in the set question first. And these questions is hard just to do one without doing the whole thing. So let's just focus on some of the data in the set question. Let's start with this one, and then we can come back if we have extra time to do other more easy question. Yeah. Okay. So this is talking about, okay, you have a fly that is, see, all this reading, unfortunately, is trying to slow you down. So you got this parasitic fly that infects fruit trees. The female lay the egg in the fruit. Larva hatches and burrow into the developing fruit. Then the adult flies emerge. So before the European colonization of North America, okay, so give me a sec. Let me see if the office emailed me yet. Not yet, huh? Again, on the test, you have 60 multiple choice, okay? So you have this thing is the native species of hawthorn, right? So hawthorn is the apple tree. That's the domestic one. Sorry, hold on. This is the hawthorn is not domestic. Okay. So the domestic apple tree is not native to North America, but it's imported. Okay, actually it is, yeah. So this is we have the domestic apple tree. Okay, when apples were first imported from north into the North Americas, there's no evidence that this fly, the host of fly, yeah. Okay, so here could be used as host. Apples set fruit earlier in the season and develop faster when hawthorn, where hawthorn sets later and develop more slowly. All right, so we got this parasitic fly that live in apples, okay? And then these flies, they live in apple trees, okay? So before the import of apple trees and after the import of apple tree, yeah? So before they were here, there's no parasitic flies. And now they do, right? There are, right? So the parasitic fly has shown that two distinct populations evolved from original population that were parasitic on hawthorn. One eats only apple, the other infects only hawthorns, right? So let's say we have apple trees and hawthorns. The cycle of both fly populations are coordinated with their host trees. The flies of each population apparently can distinguish select males with similar host preference and reject males from the other trees. So this is sexual selection here, okay? Because we have to figure out why are there different species, two distinct population, probably evolved. So this has to do evolution, right? So there's very little hybridization, that means interbreeding. About 5% they can interbreed and have babies. So they're pretty much different species. And we're told this has to do a mate selection. Okay, so my guess is that the female from hawthorn prefer to mate with males from hawthorn. Female from maybe apple tree mates with males from apple tree. Yeah. Okay, so number 20. The answer is A for this one. The emergence of two populations occur very quickly because of A. The only clue is the apple trees were imported by the settlement. Okay, so not the fly, the apple tree were imported right here. So prior to the European colonization, then apple tree were first imported, right? Before that, there's none, and now they are, right? So the best answer is A, based on the readings. Kind of like SAT verbal reading, very stupid, yeah? Number 21, okay? Number 21, the answer is B. Initially, there were isolating mechanism that is likely have been the most important. Which isolating mechanism prevents the gene flow? Okay. Why is the answer temporal isolation? Okay. So the reason is that, okay. So if you look at the clue here is that, okay, right here. You see that? Apples tree set earlier in the season develop faster, and hawthorn sets later and develop more slowly. And then there's a mate selection based on the host preference. So That means that if the males were feeding on hawthorn, they have to breed later on in the season. If the male feeds on the apple tree, they would have to mate and develop earlier, reproduce earlier in the season. So this is a temporal isolation. There's no mention on the gametes, sperm and eggs that are incompatible, right? Or that the hybrid dies, right? Or that physically and anatomically males and females are different. So the best answer is B, right? Number 22, mating between individuals from two populations of the parasitic fly produce a hybrid that looks healthy and have normal lifespan. However, the eggs hatch less than those flies from the other two. So what is the isolating mechanism? So that has to be with the reduced hybrid fertility, right? The hybrid is not 100% fertile, right? So that's common sense, number 22. All right, so number 22. And is this, so that's it, data in the set, yeah? Let's look at another data in the set, and then we'll come back to do the other one. Let's look at this one. So here's an experiment. We got 5 questions here. You subtract at 1. In a classical experiment, investigating the gene expression. So expressing the gene. A solution with equal amount of, we have rabbits alpha hemoglobin mRNA. So this mRNA will produce hemoglobin protein in rabbits. And we also have beta hemoglobin RNA, mRNA. So that will produce the beta hemoglobin. So it turns out hemoglobin have alpha subunits and beta subunits. So there's 2 alpha, 2 beta. Together is a 4 subunit molecule that carry oxygen in your red blood cell. So anyway, mRNA produces the protein. So they code for subunits of protein found in red blood cell, which is injected into newly fertilized egg. So if you inject this mRNA into the fertilized egg, then the fertilized egg should ideally produce those protein, right? Hemoglobins. All right. The question is that the injected mRNA was not degraded. Okay, so it didn't break down. Tadpoles that developed from the injected egg were dissected into 2 fragments. So here you see the tadpole that comes, we cut them into the notochord, muscle, and nerve. And we have the other fragment, which is all other tissues. And what we do is that we isolate the protein and we look for on a gel electrophoresis. What this tells me is that the difference between these two is very little. Whether you are looking into the notochord muscles and nerve or any other tissues, you have tubulin, you have both beta hemoglobin, and you also have alpha hemoglobin. I don't see any difference. So stupid, right? So why is that? Okay, so number 27. Number 27, the answer is D. So rabbit, so the question is that the observation that rabbit mRNA was successfully translated into frog. So how does that work, right? Well, because we have genetic code is the same. Same mRNA, same codon, same amino acid. So D is the best answer. Yeah. Does that make sense? Okay. And 28. 28 has to do with gel electrophoresis. Now, gel electrophoresis separates molecules like DNA, RNA, or even proteins by size and charge. So if I were to ask you which protein is the smallest, would be alpha hemoglobin, then beta hemoglobin. Tubulin is huge. So they're separate by size and charge. The smaller one move faster, okay? All right. So number 28. The answer is B for boy. The result shows what? What is the support? Well, the answer is B. Cells from different tissues share a common ability to use the mRNA, the genetic material, from a foreign source, right? From the rabbit to produce protein in the frog. So B is the best answer. Okay. Now, A is wrong. If they're saying that you resulting cell losing ability to synthesize protein, seems like they're all the same, right? Okay. And then let's look at 29. Which of the following is the best justification of why rabbit hemoglobin protein were found throughout the tadpole? Okay, so how come it's everywhere, right? So 29, the answer is D. The mRNA injected in the fertilized egg is able to distribute to every single cell's cytoplasm. So every single cell will have the mRNA and therefore have the rabbit hemoglobin protein. Yeah. So that was the assumption here. Now, you cannot say A. You cannot say that some of them are more stable than frog. There's no evidence there. And none of them are degraded, by the way, right? And you cannot also say that rabbit hemoglobin are synthesized more effectively than frog hemoglobin. There is no frog hemoglobin here, right? Okay, let's look at 2930 now. Which of the following conclusion is most consistent with the results of this experiment? Number 30, the answer is C. So the only detail that we know is that the hemoglobin have different size, charge, and shape because alpha is smaller, move further, right? We know that, okay? And we don't know anything about the RNA sequences, so you cannot say that the rabbit mRNA has different sequences than frog mRNA. No. We also don't know the volume of the blood. Doesn't make sense, right? And we don't know the synthesis rate, how fast beta hemoglobins are produced. We don't know that. Okay, number 31. Given equal amount of different mRNA injected into fertilized frog eggs, which of the following conclusion is most consistent with gel electrophoresis? So 31 is A. Okay. 31 is A. Let me see. Given equal amount of different mRNA, beta is more efficient. Why is that? Because beta is thicker. Thicker means you have actually more protein. You see both columns, the beta hemoglobins are thicker, meaning you're making more beta hemoglobin than you are alpha. Alpha is thinner, so you have less protein. So that means that if you have the same amount of mRNA, right, that's what we're told, equal amount of the mRNAs, you get more beta than you do alpha. So A is the best answer. Efficiency depends on how much protein, the width of the band. Okay, so 31 is A. All right. Let me find the next data in a set question. Usually you have a lot more data in a set, but this test, I don't see a lot. Are we done here? Oh, one more. Let's look at this one. Okay, so 41 to 45, we're talking about photosynthesis, yeah? So photosynthesis can be measured using chloroplast suspended in a solution of DCPIP. Okay, that is a dye that can accept electron from the electron transport chain. Okay, so transfer of electron to DCPIP decreases the absorbance. Okay, so, well, that's a lot of information here. So this means that you don't have to memorize it. So here's an example, what I want you to see. Yeah, so if you put in a test tube, and we can test photosynthesis by using a suspension. And what do we put here? We put chloroplast. We don't have to put an actual plant. Okay, we can put chloroplast plus this dye, and this dye can change color. Okay, this dye is DCPIP. Okay, now what color doesn't really matter. What matters is the absorbance. So when the dye pick up the electron, then the absorbance goes down. Okay, so that means photosynthesis is occurring. Okay, so absorbance, this dye actually go from blue, which you don't need to know, but I just wanted to show you what exactly it means. Yeah, so the dye starts with blue and then becomes colorless as it absorbs the electron. If the dye absorbs electron, that means photosynthesis must be occurring. That means electron transpiration is occurring. Then it's going to turn colorless. Okay, so the more colorless the dye, the faster the rate of photosynthesis. So we're correlating absorbance. So when the dye is blue, you have lots of absorbance. Yeah, blue means high absorbance. The deeper the color, the more concentrated the color, the greater the absorbance. Okay, so the darker the color, the greater the absorbance. Of course, colorless means you have low or none, no absorbance. Okay, so the faster the color decolorizes, the faster the rate of photosynthesis. Okay, so that's what the absorbance is. So you can see that if you put the plant right next to the lamp, you can see the absorbance drops quickly. That's good. That means lots of photosynthesis, right? Initially, very blue, right? So the experiment is telling you the same thing. If you put plant that is right next to the light, is able to absorb light effectively, then it's going to do a lot of photosynthesis, and you can see high absorbance. In this case, high is 0.89. That's the absorbance. And now the absorbance now becomes almost none, right? So that means lamp sample number one has lots of photosynthesis, lots photosynthesis. Yeah. Again, photosynthesis, the faster you do photosynthesis, the more quickly absorbance drops. Okay, photosynthesis. Okay, does that make sense? So in other words, I can do a little bit math. I can find out the change in absorbance divided by the change in time. Then I can find out the rate of photosynthesis. Okay. And if you were to plot this right here, yeah, if you plot it, y-axis the absorbance and x-axis is time, you should get a slope, right? So let's say you have, if you plot the first plant, sample one, you will go from, let's say we have 2, 4, 6, 8, 10. I start from 0.9. Okay, so these are the absorbance. I'm just doing a very, very, very quick and general plot to show you that if this is actually a lab question, this could show up on question number one, okay, which is lab. Okay, so absorbance went from 0.8 to 0.6 to 0.4 to 0.2. So you get the idea. And if you actually plot this, 0.89 is somewhere here, and 0.6 is somewhere here. And then you have 0.3 somewhere here, and then you have almost zero. So this slope of this line is actually your rate, okay? So the slope, which is changing y over changing x, is the rate of photosynthesis, yeah? You can actually plot this. And then you cannot use a bar graph. You have to use a scatter plot or a line graph, okay? Draw a line of best fit. Do not connect dots, yeah? So that's what this is, all right? So number 41, okay? 41, the answer is B for boy. Which of the following provide the best indication that light is required for activation of electron transport chain? Well, you can look at B is the answer, comparing results from 2 to 3. Why is that? If you look at 2 and 3, 3 is dark. How do I know 3 is dark without light? Because it's wrapped in foil, right? You have an asterisk right here. So if you look at 3, they're the same distance from the lamp, but without light, absorbance remain constant. There is no photosynthesis. But if you are exposed and photosynthesis occur quickly as absorbance drops quickly. So you want to look at sample 2 and 3. So 3 would be called the negative control. Does that make sense? Yeah. So negative control means you're not going to see photosynthesis, right? And you can argue 1 will be the positive control because you are seeing a bunch of photosynthesis, right? So it depends what you compare. So 41, the best one shows you that light is required. For that is actually B, comparing 2 and 3. Okay, let's look at 42. 42, the answer is D. So the question is, which of the following can be concluded from the data? So D says light from a lamp can substitute for sunlight for simulating the photosynthetic process, chloroplast, okay? Why not A? A says chloroplast must be sustaining buffer to functional properly. That may be true, but that's not what the purpose for the experiment is, right? And it says optimal temperature. We don't have the optimal changing temperature. Everything is the same temperature, okay? Because we have to hold the temperature constant, right? All right, so at room temperature it says here. So that's not it. So that's pretty easy, right? 43, if additional sample containing the dye was placed 90 centimeter from the lamp, which prediction would be most consistent? Well, 90 is actually even further than sample number 6, right? So 90 will be further from sample 6. So the absorbance will change even less. So if you look at the answer for 43, the answer is C. the change in absorbance over time would be less than any other sample, okay, except the control, right? So that would be the best answer, C, all right? Okay, and then 44, which of the following description of photosynthesis best explain the result of the experiment? So remember the dye has to do with electron transport. So they absorb electron from light reaction. Remember, light reaction is the electron transport. Okay, we said earlier today, okay, that this photosynthesis, the light reaction is like this box, right? So water goes in, that is the source of electron, oxygen comes out. So water is the source of low energy electron. It gets transported by electron transport chain. Final electron acceptor is actually this NADP plus will pick up those electrons and become NADPH. This is why photosynthesis produces NADPH. It also produces ATP by chemiosmosis. So the dye replaces this guy, the electron carrier. This is what the dye replaces. Replaces NADP plus. So this dye is used here, DPCIP, whatever that's called. Okay, so that's the dye. When the dye absorbs electron, it will go from blue to colorless. Okay, so this is the DCPIP or DPCIP. This would be colorless. So every time there is photosynthesis, the dye decolorizes and loses blue. That's why the absorbance drops. Okay, so that's the purpose of this dye. Okay, so what is the description of photosynthesis? Explain the result. 44, the answer is A. Availability of electron transfer depends on light energy, for sure. Yeah. And you can see that B doesn't make sense. It's talking about the dye move across the membrane. It has no, we don't know. The dye just changed color. We don't know where the dye is, right? It doesn't move the membrane in less than 60 seconds. It doesn't make sense. And it said chlorophyll degrades rapidly. It doesn't make sense. The dye does not break down chlorophyll. Okay. And it says this can only be used to measure low light level. That's not true. We have a 15 centimeter that's right next to the lamp, right? And it's working. All right. So that's that. Number 45. What is the scientific question that could be investigated using a similar dye experiment? Okay. So 45, you got to read through all of them. B is the answer. What wavelength of light is the best? Well, that's a good one because I can change we were using white light, right? That means light that has all the wavelength. We're using white light. But what if I use green light? What if I use purple light? What if I use yellow light, right? So you can change that and then you have a good experiment that do I still have photosynthesis if I use green light? No, right? If I use red light, yeah, okay. So that would be the best answer. But this has nothing to do with carbon dioxide requirement. It has nothing to do with C. We don't know which molecule accept the activated electron from the dye. Okay, we don't know. There's no way the experiment can tell which molecule actually accepts the electron. I'm telling you, it is this molecule here, but we don't know from this experiment, right? And there's no information on gene expression. Alrighty. So there's that. Okay, is there any another data in the set right here? So this is 48 to 51. Oh, so this is pretty long. So looking at let's look at this one for a sec. Yeah. Okay. And you guys okay? Yeah. Okay. I want to go over your mistakes, but data in a set is usually the most difficult. So I want to spend some time on this. So let me take a picture so that we can at least look at everything from one screen. Okay. And the thing about the paper test is that you're able to do that. We have the code down table. Okay. And I also have this. So this allows me to kind of look at the question both perspective. Yeah. I have that and this. Okay. So let's look at the question. This is looks like it's a pretty good question that relates genetics to codons to gel electrophoresis. Yeah. Looks pretty good. Okay. So the question says the following figure display data collected while studying a family. Some of the members have sickle cell anemia. Great. Well, not great. Yeah, but sickle cell anemia is a mutation in the hemoglobin gene. So that's important. So you should highlight that. Okay. So HBB, if it's mutated, you get sickle cell anemia. But we don't know what kind of inheritance. Is it autosomal dominant? Is it autosomal recessive? Or is it sex linked, right? These are the three most common ones. And they tell you that there are two alleles. Of course, every gene has two alleles. So we have the HBA codes for the wild type, and HBS codes for the sickle cell. Okay. So dominant, recessive. Instead of call them dominant, recessive, they want to use HBA for the dominant. So that's probably the dominant. Wild type is usually the dominant, not always. And HBS is the other allele. So you get either you're homozygous or heterozygous, right? So you can be HBA, another HBA, you're homozygous. You can also be heterozygous. So you have one HBA and the other one is HBS. Or you can be homozygous for HBS, like that, okay? And sickle cell anemia, you should know, is most likely autosomal recessive. So this would be the diseased individual, all right? But let's say we didn't know that. And then we continue reading, right? Genetic testing provide insight to the inheritance pattern. So we need to be confirmed with that, okay? So if you look at the pedigree here, you can see that, so instead of calling this, I'm just gonna use our regular method, like big A, this is big A, little a, and the SNS is little a, yeah? So it kind of, this should be autosomal recessive. So this disease is little a, and this is heterozygous. So you have the disease allele, and this would be heterozygous, and this is heterozygous, and this is disease. And you can get two heterozygous one, and then you can get a diseased individual. And this one is big A, or big A, little a, we don't know. So I wrote down all the possible genotype, assuming this is autosomal recessive. Yeah, that is the most simple way to look at the pedigree. And it does not conflict autosomal recessive. So I'm going to assume it is autosomal recessive. Yeah, okay. All right. So, and then look at this DNA sequence, figure 2. Okay. So the DNA sequence from the wild type. Okay. So this would be the wild type hemoglobin A. So this is the dominant allele. I don't know what the sequence is for the recessive allele. I just know the dominant one. Okay. That's what it is. And DNA is double stranded. That's why. Okay. Now look at the template. Template is what you transcribe. Okay. So from the template, that is how you get the mRNA codons. Okay. So here's the mRNA, 5 prime. You have GAC, so this would be CUG. This is the mRNA. You transcribe always the template, not the non-template, yeah? And TGA becomes ACU, and GGA becomes CCU, and we have CTC, so GAG, GAG, and TTC is AAG, and finally AGA is UCU, and that's the 3 point. So this is my mRNA, and I think that's why we're given the codon, so I can actually construct my codons, right? So if you look at the CUG here, CUG is Liu, so this would be after translation leucine, and ACU, so ACU is THR, threonine, CCU. So CCU is PRO, proline. And then GAG. Did I write it right? GAG, GAG, right? So GAG, I see GLUT. So I have two glutamine. And I have AAG. So on the chart, make sure you know how to read it. Just AAG is lyse. That is lysine. And last one, UCU. And you can see UCU is SER, which stands for serine. Yeah, so that's your protein. Okay, do you need to do this when you do the test? No, you read the question first, okay? All right, and then say here, number 48, based on the data in figure 1. So figure 1 is this pedigree, okay? Which of the following best describe the genotype of individual members? Okay. So as A says, all affected individual has at least one dominant allele. That's not true. Okay, that would be autosomal dominant, right? And 48, so A is false. If you look at B is the answer, yeah? It says, healthy individual may possess one mutant copy, the HPS. Well, that is true, right? Because, for example, healthy individual, like this one here, and also individual 2 is healthy, right? But because it's hidden heterozygous, right? So you can have the diseased allele even though you're healthy. So B is the best answer, all right? And if you look at C is wrong. 4 and 5 must be heterozygous. No, 4 is homozygous. 5 is homo, right? So let's look at 49. So 49 says the HBS allele, which causes sickle cell anemia, results from the mutation in DNA sequence. Okay, so if you look at figure 2, it says that produces a valine in place of glut, right? So do you see our valine in place of a glut? So that will be somewhere here. I'm going to highlight it. So glut, this is the normal, okay? So again, this is normal, which is HBA, but if it's sickle cell disease allele, which is HBS, they're saying that you're going to get a valine in place of glut again. So what would be your sequence? So you know it has to do with GAG, which is this portion here, CTC, right? So this portion must change. Everything must be the same. So the question is, which of the following mRNA is derived? So first of all, this has to be mRNA. So you have to transcribe the DNA into mRNA. So I have to compare to this guy here. Okay, this is my mRNA, yeah? So I know it has to start with CUG. You see that? The only thing that starts with CUG is D. There's nothing else that starts with CUG, right? Okay, so 49, by elimination, you know it's D, right? First of all, A is not an mRNA. A has T. T is DNA. C is not mRNA. So you can eliminate A and C right away. And just translate the first, transcribe the first nucleotide, you know it should be CUG right here. Because GAC becomes CUG. So it cannot be any other choice other than D. All right, so this is a transcription translation question. Let's look at next one. Next one is number 50. So 50 talk about restriction endonuclease. This is the molecular scissors, restriction enzyme. They will cut DNA or RNA, yeah? So restriction enzyme is fancy way. The fancy way to say restriction enzyme is restriction endonuclease, and that's the name of it. And they're found in bacteria that cuts foreign DNA that enters them. Usually those foreign DNA come from virus. So you want to cut them to protect yourself. But in genetic engineering, biotechnology, humans take those enzymes, use them as molecular scissors. So you can cut the DNA, then you can insert a DNA to do genetic engineering. Okay, so anyway, they tell you this. This recognizes this right here. So what does that mean? Okay, if you look at the first sentence, it's going to cut the DNA at this site, okay? So that means every time you see this sequence right here, it's going to be cut. That is essentially what that means, okay? What is the sequence that triggers a cut? Is this right here. So there's 6 nucleotide here. N can be anything. So for example, if you see CCT and N can be anything, AG, then it's going to cut. Okay, that restriction enzyme will cut right here. Okay, so let's say the 3' will be GA, whatever this is, and TC. Yeah, so this is where it will cut. Okay, so it's going to cut right here. That's what they're saying. Yeah. Producing separate fragment. Which of the following best explain the bending pattern in figure 4? So now we look at this now. Okay, so the idea here is this, that you can tell who's homozygous dominant, who is heterozygous, who is homozygous recessive by looking at the gel electrophoresis for those three individuals, okay? So if you look at the gene, how do you know who is heterozygous? It's always the one with most fragments. Can you memorize that, please? Always, okay? So let's say, for example, an individual 3, yeah? Heterozygous is always individual 2, always, okay? So this would be your heterozygous, which means it's big A, little a. And in my notation is big A, little a, but actually is HBA, right? Remember that's the wild type. And also HBS. It's hard for me to write for some reason. And then you also have HBS. That's what heterozygous mean. Okay, so that would be always the individual with the most fragment, heterozygous, period. You just have to memorize it. It's so easy, yeah? And the other two are homozygous. But we don't know which one is which to say, yeah, for the least. So this would be, oh, I can't write this. So I'm just going to write big A, yeah? So this would be big A, for example, or an individual should be little a, okay? If the big A has no cut, no site, and little a has one site, okay? So let's say this fragment is 30 nucleotides long, this is 70 nucleotides long, this would be the 30 nucleotide, this would be the 70 nucleotide, and if you're not cut, you're 100 nucleotides. So this would be 100, and this would be 100 here, and this would be 70 here, this would be 30, right? Remember, small fragment is there. So what does that mean, cut, right? So if you have the restriction site, that will be cut by this particular restriction enzyme, the enzyme endonuclease. They call this MST2. Then it will cut right here. So do you see that if you're a homozygous dominant, you will not be cut at all. So you will have one giant fragment. And if you are heterozygous, you're going to be cut into 3 fragments. So these are the 3 fragments. And if you're homozygous recessive, like if you are 2 little a's, then you're going to be cut into 30 and 70. You're not going to have 100, right? Because you cut. So that's why individual 3 is homozygous recessive, right? So the heterozygous is going to have all the fragments possible. Okay, so that's what the bending pattern is. So which statement explains that? Number 50, the answer says A. So the HBA contains the recognition site. Okay. So then, so this is the normal one. Okay. So it has a recognition site. So yeah, so they actually reversed this. Okay. So in this case, they reversed this. They're saying the normal one has a site. So that'll be cut into 2 fragments. Okay. So in this example, they reversed it. Okay. What I show here. So instead of drawing that again, instead of erase and draw, I'm just going to show you this again. Yeah. So the HBA is the normal one. Don't forget. If the normal one has a cut side, then I have to draw the big A has a cut side. So let's say this is 30 and this is 70. Yeah. Yeah, like here. Okay, 30 and 70. And little a will no cut side. So it will be 100. So that means the one that did not cut, this will be the homozygous recessive. So these are the people who have sickle cell anemia. And this is reverse. So this is the big A. They have one side, so they'll be cut into 2 fragments. If you have one cut side, you're going to get 2 fragments. And again, individual 2 is heterozygous for both. So that's why you have most number of cuts. Okay, so they just reverse what I have here. So A is good answer. Good. Now B is wrong. If you have 3 recognition sites, wouldn't you be cut into 4 fragments? Kind of weird, right? So then this would not be the pattern, right? So that's wrong. And C is just a false statement. You cannot have 1 copy of the gene. That means you're haploid for the gene. You have to have 2 copies, 1 from each parent. So that's just wrong. And D is wrong. You're saying that there's 3 different alleles when you're told there's only 2 alleles. So you can do it by elimination. All right, let's look at 51. Possessing a single copy of the recessive allele, right, which is HPS, provides some resistance. That would be the heterozygous advantage. So if you're heterozygous for sickle cell anemia, you're resistant to malaria, which is caused by this organism, right? So which individual have resistant. So 1, 2, 3, or 4. So you look at the pedigree. Who is heterozygous, right? So we already established that here when we analyze the pedigree. And I have heterozygous individual. Uh, individual 2 must be heterozygous, right? Individual 2 must be heterozygous. Okay, not 3. 3 can be both hetero or dominant, homozygous dominant. So individual 2 is the answer. Okay, so that would be B for number 51. Okay, so that's all the question here. Let's see. We have a little time, so let me just go over maybe a few that you guys have question on. Do you know anybody want to volunteer? Yeah, which question? I I might be able to see from your scoring chart. Okay. Um, okay. I have some of you guys' score. Yeah. So maybe I'll just pick some then. So so the let's do another data. Oh, 10, 11, 13. We didn't do this one. Okay, let's look at this one. Okay, so this is all the data in the set. So you have changing population of 2 species of beetles. Okay, so you have the answers, right, guys? Make sure you have the answers so you can also go over the ones that we couldn't finish. So make sure you understand them. Yeah. Okay. So we have beetles. All right. 2 species. I'm not going to try to pronounce them. So one is confusum, one is castadium, right? So ca, cast and con, right? Conf and cask. Okay. The two there's no parasite if you look at the open circle, and the closed circle is parasite. Okay, so each data point is an average population from 10 dish cultures of equal size and same amount of food. So I'm looking at the number of beetle, okay, over 900 days. So here is the confusum, okay, it's the first species, and looks like it's the same. Okay, whether there's parasite or not, the average number of culture is roughly the same. But figure 2, when you look at castadium, that is different, right? So without parasite, they seem to fluctuate, and with parasite, it's like that, yeah. So there is some difference here. Okay, so we don't know why. So let's see what the question wants. Okay, so number 10, the answer is C. Under which of the condition is the observed number of beetle per dish the greatest? So C, if you look at castanium, which is the bottom one, 100 days. So bottom one, 100 days is, are we looking at parasite? With parasite. So with parasite, 100 days, I'm looking at about 30, right? But if you look at the other one, so you got to go through all choices. So C is 30. What about A? Confusum at 500. So if I like a confusum at 500, is like less than 20, right? Okay. So no, right? So you got to go through all of them. So this is extracting information from data. So the answer is C. It's about 30. Okay. Number 11, the data over the duration of the experiment provided the strongest support for which of the following conclusions regarding the effect of the parasite on these two, right? So that would be the difference between these two, parasite without parasite. Okay, so it seems like the bottom one, the castadium, grew better when there's no parasite. And if you look confusing, the top one doesn't seem to be bothered, right? Whether you have or you don't, they have the same growth curves. So that's our own conclusion. So number 11, you got to read through all the choices, right? If you look at A is wrong. Confusum, the top one, is adversely affected by parasite. Well, that is not true. They have the same growth pattern. So A is wrong. Nope. B is the answer. You can see that castadium, the bottom one, is adversely affected by the parasite, but the top one is not. That's the conclusion. The top one is not affected adversely. The bottom one is, because it grew better without the parasite. Easy, right? So look at the data and make sense out of it. You have to read through every choices by elimination. Elimination is the key skill in AP Bio. And number 12, in figure 1, the difference between the curves can be attributed to which of the following? So it's the parasite, right? Okay, so it would be the effect of the host on its parasite. Well, hold on, that's not. It's the effect of parasite on the host, so no. Okay, so in figure 1, there's all very similar, right? But there's still tiny variations. So that would be D. the natural variation among the population. So is that the parasite have no effect. It's just naturally they're a little bit different, but they're similar enough for us to make a projection. So D is the best answer. The difference in figure 1, okay? All right, number 13. If you continue for 500 days, the population of the Castanian will be the bottom one, okay? With parasite will most likely stabilize what? Okay, so with parasite is the dark one. So if you continue, it's going to stabilize about 10, isn't it? It's already stabilized, right? Since 400 days. So if you last for another 500 days from 900 days, it's gonna probably stabilize or even go down. So it's gonna be 10 or less, right? So 10 or less, what do you think then? Is it A or B? Number 13 is B, okay? Because it seems to stabilize, so you shouldn't drop from this point on. The trend is stabilization, okay? So B, 10, Beetle would be the best. Alrighty. So okay, so guys, well, I guess we'll stop here. Now there's also some of those evolution cladogram questions. They're actually quite easy. Sorry, I didn't get to go over all the questions. I was hoping to spend more time reviewing the free response because multiple choice I think is a little bit easier. But you guys should have the answer. Let me see. To these questions. Yeah. Okay. If you don't have the answer, please ask the office for an answer and look over the mistakes you made. And sometimes you can figure it out on your own. Yeah. All righty, guys. So let's stop here. I thank you so much for coming in. Yeah. For so long during spring break. Appreciate it. I'll see you guys later. Yeah. Okay. All right. Bye, guys. Bye, guys. Thank you, guys.